Dr. Green is a world-renowned scientist who co-pioneered the Human Genome Project. This keynote lecture takes place on an occasion that Dr. Green represents USNIS to receive the Prime Magnitude Award 2017 for the achievements in medicine. The Department of Medical Science has a mission to become a leader in medical science research and innovation of Asia. Therefore, this year, CMSC together with Thailand's Research University Network and other national agencies will run the National Strategic Project on implementation of medical science and precision medicine. Hence, today is a great opportunity of our project collaborators and researchers to meet with Dr. Eric Green, whose vision leads to many international collaboration projects that drive a new era of precision medicine. And now, this is my honor to invite His Excellency Clinical Professor Emeritus Dr. Piyatapon Satayakon, Minister of Public Health, to give the welcome remark to our keynote lecturer today. Thank you.
Dr. Eric Green, distinguished guests, ladies and gentlemen, it is my great pleasure and honor to welcome Dr. Eric Green, which we have met last night. He is the director of the National Human Genome Research Institute, National Institute of Health, the United States of America. Dr. Green represents the NHGRI as this year's awardee of the Prince Milan Award 2017 in the field of medicine for the achievement of the Human Genome Project. The project was launched in 1990 and was a collaborative effort from 20 institutes in 6 countries, making it the world's largest collaborative biological project. The project aimed to tackle the fundamental elements of life, which is to decipher the DNA code of a human genome. After 13 years, the project came to success, and the Human Genome Code was since publicly available. Complete understanding of the human genome has led to discovery of treatment of genetic diseases, cancer risk prediction, and tailoring the novel targeted therapies. The outcome of this project not only improved the quality of life, but also revolutionizing the ways in practicing medicine by incorporation of the genetic information in the prevention, detection, and treatment of diseases. We now know this project as precision medicine. In addition, the scientific community has seen the leap of DNA sequencing technology that makes personal genome sequencing possible today. This led to sequencing efforts for millions of individuals. Okay, without further ado, I would like to welcome Dr. Eric Green to give us a keynote lecture. I wish that by learning from Dr. Green's experiences today, Thailand will move forward on implementing precision medicine into our healthcare system to benefit our people. Thank you.
Good morning, everyone. It's an honor to be here to give this talk to you, to visit from the United States. As you heard, I am here to accept this great honor, not for myself, but really at the ceremony later today, but really for the thousands of people and scientists from 6 different countries that work on the Human Genome Project. What I thought I would do today is to really tell you about this journey that began with the Human Genome Project, that is finding its way into medicine, that is expanding the frontiers into new ideas for how to practice medicine more precisely. And it really is this journey that grew out of this new field of genomics. Now, let me start by pointing out that this field of genomics that I represent is very young, much younger than biochemistry or cell biology or physics. In fact, the word genomics was only put into the scientific literature for the very first time in 1987 in the lead editorial of a new journal called Genomics, where the editors described this newly developing discipline of genome mapping and sequencing, for which they adopted this term genomics. A genome is all the DNA of an organism, and the idea of comprehensively studying a genome is really what genomics is all about. By the way, 1987 was the year I graduated medical school and also graduated graduate school. I had an MD and a PhD, which means never once during medical school or graduate school did I hear the word genomics because it didn't exist. So in my professional education, I never was introduced to the word genomics because it was such a young, evolving discipline. But when I had the opportunity to then do postdoctoral research as a resident of pathology, I was attracted to a laboratory that was one of the first to actually begin using genomics for studying human DNA. And I was very excited about the opportunity that was being discussed in the late 1980s to participate in this large, international, team-oriented effort called the Human Genome Project. And I found it so exciting and so compelling that I joined the project literally on the first day on the front line as a postdoctoral fellow and participated in the project from beginning to end. The project was originally thought to take 15 years. We were successful and did it in only 13 years. And the major goal of the Human Genome Project was to read out the roughly 3 billion letters, or in other words, bases, that are in the human genome. In other words, decoding the human blueprint for the very first time. Remarkably, that goal was achieved, and we look back on this now 15 years later and marvel at the fact that we were successful at doing something that when the project began, we weren't certain was even going to be successful. But rather than tell you about the details of the Human Genome Project, what I thought you'd be much more interested to hear about is what has happened in the 15 years since the Human Genome Project and what's going to happen in the future. So that's what my talk is going to focus on. Let me try to talk about the highlights that we've seen. Genomics has now been around for 3 decades, from 1987 to 2017 or so. What have been the big highlights in genomics in its first 3 decades? Well, I told you about the first of these. Highlight number 1, we sequenced the 3 billion letters of the human genome for the very first time during the Human Genome Project. What happened next is truly remarkable. Because what happened in genomics is that the tools of analyzing DNA became widely applicable to many, many different areas. And in fact, every one of these areas has been significantly influenced by genomics. In fact, I would contend some of these areas are of great interest to this ministry and are incredible importance, especially infectious agents, for example, and microbiome and so forth. But I'm not going to talk about any of these. There are other experts you should get in to talk about these areas of genomics being applied. What I have expertise in, what my institute works on, are the applications of genomics to health and disease and medicine. And in particular, having completed the Genome Project 15 years ago, what my institute's goal was then to understand how to use the human genome's basic information to eventually be able to treat patients differently, recognizing that every patient is a little bit different. Every one of us has a blueprint that's a little bit different. And if we can only use genomic information about individual patients, we would have a better way of practicing medicine, more tailored to the individual patient. This is embodied by this emerging field of genomic medicine. Basically, the use of genomic information as part of clinical care. This is what we aspire to do in genomics now, coming from the Genome Project to eventually realizing the use of genomic information routinely for patient care. Now, this is a journey. It will not happen quickly. It began 15 years ago when the Genome Project ended. That was the starting line for this. We had in front of us the first sequence of a human genome, and we knew that if we pursued this journey, that maybe one day we would change the practice of medicine. But of course, that wasn't going to happen in one step. It wasn't going to happen quickly. It was going to involve multiple steps. We don't even know what all the steps are. But we knew that if we assembled many, many people from many different disciplines, and we all had them running in the same direction, sort of like a marathon, that someday we would be able to realize genomic medicine. And so this was the path that we saw going forward from the completion of the Human Genome Project. What were the other highlights that began to move us down that path, step by step, getting us closer and closer to changing the practice of medicine? Well, I would contend that the second greatest highlight in the first 3 decades of genomics has been in our ability to develop cheaper and cheaper methods for sequencing DNA. In fact, we have gone from having the ability to sequence DNA at one price and now have seen it go down nearly 1 million-fold since the end of the Human Genome Project. Let me give you a little bit more information. That sequencing that first human genome by the Human Genome Project involved basically laboratories that were twice the size of this room. There were dozens of them in 6 different countries. It involved hundreds and hundreds of people working for many, many years sequencing human DNA and eventually being able to put together the first sequence of the human genome. By the way, they were using instruments that were about the size of a refrigerator at the time. But we were successful, and we sequenced the first human genome, but it cost about $1 billion US. What we said we wanted to do the day the Genome Project ended was we need to reduce the cost of sequencing the human genome. We need to remove 6 zeros from that figure by developing better and better methods that would eventually produce a $1,000 human genome or less. We picked $1,000 because $1,000 US is a reasonable cost for a clinical test such as a radiographic test or many laboratory tests. We really didn't know how we were going to do this, but we were able to be successful by attracting very smart scientists, and meanwhile, various companies got formed that also got involved in trying to develop new methods for sequencing DNA. And guess what? We're essentially there. This has been written about in the scientific literature, and we've sort of kept track of this through various graphics, such as shown here on a logarithmic scale. In green is the precipitous decline in the cost of sequencing DNA since the introduction of these new methods that you've probably heard about called next-generation sequencing methods for analyzing DNA. But what I really want to point out is it's not just the cost, it's also the speed at which you can actually sequence DNA. What I didn't tell you precisely, but I'll tell you now, is that first human genome sequence cost about $1 billion, but it also was 6 to 8 years of actually sequencing DNA to produce one human genome. That is not a clinical test, too long. What we now are able to do, using instruments that would fit on this podium, and in fact one of the instruments I just saw in an adjacent building on this campus, is using these new ways of analyzing and sequencing DNA for something that is getting very close to $1,000, and in some people's hands is actually less than $1,000. In about a day or 2 or 3, you can generate a human genome sequence. And so we have seen this precipitous decline of nearly 1 million-fold in the cost of DNA sequencing. By the way, I'm not worried about this anymore, because whatever methods that are available now, they will be supplanted, they will be replaced within a few years with newer methods, and yet newer methods and newer methods. The one method, just to keep an eye out, because I think it's very exciting, is this new method of nanopore sequencing, which involves basically pulling DNA through very small pores and membranes and being able to monitor the electric current such that the different layers of DNA give different current values that allow you to figure out exactly what letter is being seen at that moment in time. This instrument can be miniaturized, very small. And so, in fact, this instrument is just now being commercialized, and we've progressed from having refrigerator-sized instruments to instruments that would fit on this podium to instruments that fit in your pocket. This is the latest nanopore sequencing instrument. In fact, this is not just the instrument, this is the entire laboratory. You don't need a laboratory. We could sequence DNA right here. The laboratory is located on this little chip. It's all you need. And you need one other thing, by the way. You need a laptop computer because this is a USB port. And you plug this into a laptop computer and you can sequence DNA. Now, this is not quite ready for sequencing human DNA. I think we'll be there in the next few years. But this works terrific on sequencing smaller genomes, such as the genomes of microbes. Am I making this up? Is this really a laboratory? Is this really for sequencing DNA? Absolutely. This exact instrument, in fact, was used in Africa during the Ebola outbreak to type the specific Ebola viruses in different villages. There was no laboratory. There was just this instrument in the laptop computer. This was used up in Antarctica to detect the microbes that existed in Antarctica. And this exact instrument was used in the International Space Station by this astronaut scientist. So this is very exciting. I believe there'll be better instruments. I just give you this example to remind you that there will be technical innovations and that simple instruments like this will be available worldwide, and I think the ability to jump in and begin to generate genomic data, first on microbes, eventually on humans, will be within the reach of all countries around the world. Oh, and by the way, the reason why I think this instrument may not be the last one is this same company claims to be having a new instrument coming out perhaps in the next couple of years where it'll just be a little attachment to your iPhone. We'll be able to sequence DNA. We'll see if this really comes to pass. But it's just to imagine where we have gone from factories to small instruments is really a testimony to the technical innovation we have seen in genomics. Well, we could sequence DNA that cheaply. We don't want to just sequence one genome. We want to sequence lots of people's genomes. We want to have catalogs about how we differ. We have seen a progression in 15 years from having one human genome sequenced to now having tens of thousands. Actually, we probably have hundreds of thousands of people's genomes have now been sequenced. And the reason we wanted to sequence a lot of genomes is we're interested in how we vary. We're not so interested in how we're similar. We're interested in how our blueprints differ from one another. By the way, across all of your letters in your blueprint, about 1 out of 1,000 of your letters differ compared to the person sitting next to you. That, if you do the calculations, about 3 to 5 million differences in each of our genomes compared to anybody else in this room. And we really want to have catalogs of that, and we want to be able to identify those differences and then begin to characterize those variants. Some of the variants are bad variants. They may confer a risk for getting a disease. Some of the variants are good variants. They might protect you from getting a disease or be affiliated with some other positive attribute. As these new methods for sequencing DNA became available, big projects got formed to try to sequence many, many people's genomes, catalog the differences, make them available on the internet for scientists to be able to study, so we can figure out which are the not so good variants, which are the variants that really don't matter, they don't have an influence one way or the other, and which are the good variants. A number of projects have been conducted worldwide, the most recent of which is a project called 1,000 Genomes Project. It was a project that was originally going to collect 1,000 people's DNA from around the world. It actually ended up collecting more like 2,500, but you can see from all different parts of the world, DNA samples were collected. Those DNAs were sequenced, and the variants were put up on the internet for all scientists to be able to access. Now, just because you have a variant, you don't know if that variant is important or not for health and disease. One way to begin to understand whether those variants are important is to understand how the human genome actually works. And here we have been methodically studying for the last 15 years the human genome sequence that we made available 15 years ago by the Human Genome Project, and we have greatly advanced our understanding about how the human genome works. Shown here is just 0.0001% of the human genome. And what we want to do is to know where are all the sequences that are functionally important. Where we have made the most progress has been understanding those sequences that directly code for proteins, protein coding genes. We now know exactly where virtually all the protein coding sequences are in the human genome. It amounts to about 1.7% of all the letters, a very small percent. And in total, it's about 20,000 genes. The human genome encodes for about 20,000 genes, but it's only 1.7% of the letters. We have come to learn in the first 15 years of having the human sequence available to us that there's an even greater amount of other sequences that some people refer to as the dark side of the human genome because we don't know how they work. We know they're functionally important, they are conserved through evolution, but we haven't yet figured out exactly what they do. These sequences, which I showed in a different color, are beginning to be characterized. And we are increasingly learning that these are the sequences that determine when, where, and how much genes get turned on. They provide the specificity to different cells in the human body using different genes in different combinations at different times. And we are learning more and more about these sequences. And then we've also learned that there's a whole other code of the human genome. It's not just the order of the G's, A's, T's, and C's, but there's also something called the epigenomic code, which involves DNA being modified with different chemicals and having the DNA associated with proteins. And all of these epigenomic marks on our DNA influence genome function. And so we are increasingly developing richer and richer annotations of human DNA, knowing where there are genes, where there are these regulatory elements, and where there are these epigenomic marks. Now, in reality, we still have a long way to go in understanding how the human genome works. Our institute has led a program called ENCODE for Encyclopedia of DNA Elements, which is basically building up a catalog of all the functional sequences in the human genome and explaining what they do. 15 years into interpreting the human genome, we now know, we've gone from knowing about that much about how it works to maybe knowing about that much how it works, and eventually we need to know about that much. So we have a long way to go, but I think we've made significant progress, and I think worldwide we will be, for the next 10 or 20, maybe even 30 years, we'll be interpreting and reinterpreting the human genome sequence. It's complicated, but it's really important. But of course, that then allows us, with knowledge of genomic variants across the human genome, millions and millions of places that we now know vary in the human population, and increasingly knowing where the functional sequences are, we are beginning to characterize the subset of genomic variants that actually influence traits. It's not all variants. In fact, most variants have no influence. But a subset of them do influence our phenotypes, our traits. And of course, that's what we're very interested in as it pertains to medicine. This involves understanding which genomic variants influence human disease. And I think the fifth highlight we clearly are seeing in genomics is the fact that we are making significant advances in unraveling the genomic basis of human disease. Now, all human diseases have a genomic influence. Some have an absolute genomic cause, and some just have a small genomic influence. In general, there are two major categories of diseases, and we have seen very different progress so far for the following reasons. One category of diseases are rare diseases. These are diseases like sickle cell anemia, cystic fibrosis, Huntington's disease. Rare in the population, but relatively simple at a genomic level, because it is a single letter difference, a single mutation, that basically breaks a gene. And that is the major reason why you get the disease. These are monogenic diseases, also referred to as Mendelian disorders after the famous geneticist Gregory Mendel. Here, because it's relatively straightforward to find one broken gene in a disease, we've made great progress. I'll give you an example. The day the Human Genome Project began, there were 61 rare diseases that we knew what gene was mutated to cause that disease. 61. Fast forward to today, we now know for 4,800 rare diseases, we now know what gene is mutated. Now, there's still a few thousand more rare diseases we haven't figured out, but we are working very hard to figure out the genetic cause of the remaining rare diseases. So we've made great progress for rare diseases. But as many of you know, especially at an institution like this, rare diseases do not fill hospitals and clinics around the world. In aggregate, the major healthcare burden in every country in the world are common diseases. These are diseases like hypertension and diabetes and Alzheimer's and autism and cardiovascular disease and mental illness and many forms of cancer. Common diseases are complicated because it's not one gene. In fact, it's usually multiple different variants in multiple different places in the genome that each contribute a little to the risk of getting a disease. And on top of it, there's a greater influence of the environment in those disorders and lifestyle and so forth. Here, we're making progress in figuring this out, but it's a much bigger problem. Yes, we are learning in some specific examples which variants are conferring the risk for the disease and how do those variants sort of influence biology to confer risk for getting those diseases. But this problem is going to be a major one for the next 10 years. Just as I think we've made great strides in the last 10 years with rare diseases, I think the next 10 years will be very significant for our understanding of common diseases where you need much larger studies to be able to. So those are 5 highlights over the last 15 years. But notice that none of them say anything about the practice of medicine. At most, they talk a little bit about disease. But I promised you genomic medicine, that's where we were heading. And I will tell you that 7 or 8 years ago, this is the point where I would end my talk, because I didn't have much more to say. But I actually, over the last 5 and 6 years, have gotten very excited that I can now talk about vivid examples of genomic medicine in action that are emerging. And I think this is quite significant, because what we are finding is that we are now in transition as we are bringing genomic medicine into focus. It is some of the earliest examples, and I think we are much, much more in front of us in terms of advances in genomic medicine than what we've seen so far. But I think these early successful examples are really providing us insights about how genomics might be truly used in the practice of medicine in the future. So I thought you'd be interested to hear what I think are some of the hot areas in genomic medicine, the earliest examples, even though there'll be much more development in each of these. I'll walk through them. First one is cancer and cancer genomics. No question, this will have significant impact in cancer treatment in the future. Let me remind you that cancer is a disease of the genome. The reason you have cancer is that normal cells pick up mutations in their genomes, and those tumors basically have cells running out of control because of changes in their genomes. But we have the ability to read out the genome of tumors just as easily as reading out the genome of normal cells, and we've been doing that for the last 5 years. Projects all around the world, dozens of different kinds of cancers, thousands of specimens have been analyzed, and massive data sets have been assembled. And we now are learning that cancer is a very different disease than what we thought it was, based on really peering into the genomes of these tumors. And it is very clear that we will change the way diagnostics is done for cancer. We will continue to have pathologists looking under the microscope and being able to visualize the histology changes associated with cancer. But increasingly, that information will be augmented by sequencing this tumor from that patient and deriving basically fingerprints of the changes in the genome of that tumor in that patient. And that will give a much more powerful view of each patient's tumor. We will have their morphology and we will have their genomic signatures, and together we will be able to make much more insightful conclusions about the exact type of cancer or the subtype or the subtype, and that will point to much more refined ways of treating cancer. It will also give us much better views about the biological pathways that are broken in different types of cancer. This is already sort of standard of care for some kinds of cancer, but the numbers will grow considerably in the coming years. Second major hot area in genomic medicine that this country should be very proud of relates to pharmacogenomics, pharmacology and genomics. We have recognized that indeed we are imprecise in how we prescribe medications. A recent article in Nature pointed out how imprecise we were. Shown here are 10 commonly prescribed medications. And it is known that for every patient in blue where the drug works, or all of the patients in red where the drug doesn't work, or in some cases where the drug actually brings adverse reactions, some serious, some life-threatening. We have also come to learn, especially in the last 15 years, that much of the differences in these blue people versus red people are genomic variants that influence the metabolism of drugs in those individuals. And so we are now at the cusp of a change in our approach for prescribing medications, where we won't just randomly pick a medication and give it to a patient. We will try to pick the right medication from a list of options for each patient based on their genomic profile. The reason I say that Thailand should be proud of this paradigm is because they have one of the best examples of actually implementing pharmacogenomics in the case of Stevens-Johnson syndrome, and knowing that in the populations that live in this country, there are particular known genomic variants that put individuals with certain medications at high risk for getting that disorder. And you now have approaches and are implementing them nationwide for being able to screen individuals before you make decisions about those medications. And so this is a vivid example, but there's going to be many others. There are already many others, and I think the list of drugs for which pharmacogenomic information is going to want to be obtained before making decisions about giving patients that medication will grow in the coming years. A third hot area in genomic medicine relates to rare genetic diseases. We always dreamed that once upon a time, if we had a patient and we didn't know what was wrong with them, let's just look at their blueprint and figure it out. But that's not so simple, because you have to know how to interpret all these different variants. And for years, we really weren't quite sure we could actually derive accurate diagnostic information from a genome sequence of a patient with a rare disease. But that seemed to change quickly in 2011. These are some very vivid, well-publicized examples that were published in the scientific literature. This little boy from Milwaukee, Wisconsin in the United States, and these twins from San Diego, California in the United States, where in both cases, these children were going to die because physicians did not know what was wrong with them. Their genomes were sequenced. Fortunately, people could figure out what gene was mutated, and fortunately, they could think of a treatment to save their life. And these children are all alive now. And if it wasn't for genome sequencing, they would not be. By the next year, Nature was predicting that genome sequencing was going to be set to alter the clinical landscape. And a couple of years later, Nature once again said that for disorders not readily explained by standard tests, could sometimes be diagnosed through genome sequencing and analysis. That sometimes is about, right now, 30 to 50% of the time. Worldwide, this is going on thousands of times every single month. And most studies show that presently, if you have a patient with an undiagnosed condition, you sequence their genome, about 30 to 50% of the time, you will figure out the mutation that is causing that disease. You don't always have a treatment, but at least 30 to 50% of the time, you know what the diagnosis is, and I think that number will get better with time. And what this has led to are major programs, including my understanding programs that are now being developed here in this country for undiagnosed cases. Many people have gone through diagnostic odysseys where the medical system has failed to deliver a diagnosis that is very tragic for the patients and their families. But increasingly, the tools of genomics are being brought to these patients, and undiagnosed conditions are being looked at through a new lens that, again, is increasing the diagnostic rate and in some cases saving the lives of these patients. The next area I want to tell you about that is hot really relates to what I refer to as the genomics of pregnancy. There's actually two stories here, one that relates to prenatal and then one that relates to postnatal. In the case of prenatal, this has been a significant advance for prenatal genetic testing, which has actually gone on for decades. But traditionally, prenatal genetic testing involved invasive methods for accessing the fetal DNA for testing. We'd be testing for things like chromosome numbers, like trisomy 21 or other chromosomal abnormalities. The problem with these invasive methods, such typically like amniocentesis, is that they're unpleasant, and they also risk the pregnancy, and they tend to be expensive oftentimes because it involves a medical procedure. And better methods for analyzing the DNA have become available for detecting things like trisomy 21. But by and large, we would love to see a better approach. That better approach came about because of these new methods for sequencing DNA. Because it turns out that all pregnant women actually shed from the placenta small amounts of fetal DNA that float around in the maternal bloodstream as cell-free DNA. And that it's extremely in small amounts, but these new methods for sequencing DNA are so exquisitely sensitive that through a simple blood draw of a pregnant mom, you can basically use this as a screening method for detecting chromosomal abnormalities such as abnormal numbers. What's remarkable about this non-invasive prenatal genetic testing is its uptake. Shown here is some data, whereas companies have formed to do this procedure, that from 2012 to 2014 approaching nearly 1 million, but in fact what's going on since 2014 is remarkable uptake around the world, such that the current estimate is that in 2018, 4 to 6 million pregnant women will get non-invasive prenatal genetic testing. And so this, in fact, is the #1 example of genomic medicine in action using these new methods for DNA sequencing. My very last, and so that's prenatal. What about postnatal? What about newborns? You know, some magazines have predicted that by 2025, maybe everyone's going to get their DNA sequenced at birth. I'm not sure that's true. We are actually studying this, and as are other countries. Is it imaginable that every newborn would have their genome sequenced and have that sequence information carried with them in their medical records as they go through life? And we will see. I think there's some important logistical challenges. It's expensive, probably can't do it quite yet. I think there's a lot of ethical issues as well in terms of who has access to the information. One setting where it is very clear this is here and now is in the setting of the acutely ill newborn. And in fact, we have researchers in the United States who have developed a very rapid method for sequencing the genome of acutely ill neonates in neonatal intensive care units. Oftentimes, knowing that this child might die in 2 to 3 days, they don't know what's wrong, they've developed a very rapid genome sequencing approach that is delivering answers about those neonates in about 1/3 of the time. And so I can tell you in the United States, neonatal intensive care units are getting ready for being able to offer rapid genome sequencing for some of the acutely ill patients that they have where they don't know what is wrong with that newborn. The very last hot area is hot, not so much because we're making progress, but hot because of the challenge associated with it. It actually turns out that we find ourselves in an interesting place right now. We can generate a human genome sequence pretty easily, $1,000 or something like that. Generating that genome sequence on a patient is very, very straightforward. But it's not really routine yet to analyze that genome sequence. So we have the ability to generate the information, we just don't yet have the ability to rapidly interpret that information. And in fact, when we sequence a patient's genome, and the very next day, we go to round on that patient, most of the time this is what we feel like as physicians. We're just not quite sure exactly what all the different letters mean and what all the different variants mean. We need to accelerate the pace at which we can go variant by variant and tell the busy physician or pharmacist or nurse which variants are important for the practice of medicine. And what we need, therefore, is a clinical genomics information system, one that integrates with electronic health records, and one that basically allow a busy nurse or physician or pharmacist to quickly look up on their iPhone or on some little electronic tablet that will immediately tell them which variants are important, whether it relates to medication or relates to some other part of their care. And We're just not there yet, but we're trying to get there. And we have major programs, I can tell you in the United States, I know similar programs in other countries, to try to develop this knowledge base of information about variants and how they relate to the practice of medicine. So this is another big challenge going forward. It's hot because there's a lot of activity, but we have a long way to go to fully be able to routinely use genomic information as part of the busy workflow of healthcare professionals. So that's what I wanted to tell you about genomics. So that's the discipline I represent. This is the discipline I've worked in for something like 30 years. But in the last few minutes, I wanted to bring you even further into the future. And I thought I should be honest with you by pointing out that all of disease is not genomics. What I have mostly told you about is the genomic contribution to diseases. And I can talk a lot about that because what we have seen in genomics in its first 30 years has been remarkable new technologies for analyzing this part of the pie chart. We have gotten to the point where we are learning about variants, we're increasingly understanding their role in health and disease. But if we're really going to better treat patients, we need to also have the ability to better understand the contributions of the environment and lifestyle and social factors to human disease. And I think the reason they have lagged behind is because they just haven't had very good technologies. I think that's going to change. I think we're starting to see the emergence of new technologies for measuring environmental factors, for measuring diet, for measuring the lifestyle. And with that, we might be able to harness a more precise way to practice medicine. This has given rise to the notion of precision medicine. And genomic medicine is just about the genome, and genomic medicine is a subset of precision medicine. The G's, A's, T's, and C's of our genome are part of precision medicine. But precision medicine broadens it a little bit and looks beyond the genome to other factors in people's lives, such as lifestyle, environmental exposure, and so forth. And so the way I like to think about precision medicine, based on what we've learned in genomics, we are interested in understanding a more precise accounting for individual variability. I've spent most of my talk telling you about genomic variability, but we should equally appreciate that there's physiological variability and that we should be able to measure that through mobile health devices that maybe measure blood pressure or maybe measure blood analytes or maybe do EKGs, not one time in an office visit, but thousands of times, and collects that information on iPhones that transmits to databases. And if we could understand that better, we would know much more about aspects of physiology in human disease. We have better and better ways of measuring lifestyle. We could measure how much we exercise. We have better ways of measuring how we sleep. We're increasingly developing technologies for better measuring our environment. And we're learning more and more about how to accurately capture information about what people eat, how much sun people are getting, whether they're smoking or not, and so forth. And so the idea is, could we collect a lot of data about a lot of people, both genomic, physiological, lifestyle, environmental data, and be able to learn what the different contributions are to human disease? And if we could do that, we might be able to have a different way of practicing medicine. We might be able to go from the way we do it today, which is mostly practicing medicine based on the expected response in the average patient, to a tomorrow, that eventually maybe we'll be able to do medical care based on individual genomic variability, where we can start to see the potential, but also environmental variability, lifestyle differences, and so forth. Simply being more precise. And the big question is, how do we get from today to tomorrow? Well, one way you can get there is through major research initiatives. And there are these kinds of research initiatives that are basically popping up around the world. In the United States, we are in the midst of launching a big program of research in precision medicine that's embodied by a program called All of Us, the All of Us Research Cohort, where we are now going to be recruiting 1 million Americans to volunteer. When they volunteer, they're going to share genomic data, lifestyle information, biological samples, all linked to their electronic health records. They're going to wear mobile sensors, Fitbit-like devices, other sensors. We're going to collect information on them, not for 1 year. We're going to probably collect information for a decade or 2 or 3. And all of this data collected on all these people is going to be shared with any scientist who who wants to share it responsibly. We will build in a lot of privacy protections, but the idea is to empower data scientists to analyze this kind of data at scale and tease out all these small bits and pieces that are contributing to human disease: genomics, physiology, lifestyle, environment. This is unprecedented, of course, but many countries are doing this, recognizing that genomic data coupled with electronic health records, coupled with these new technologies, which will get better and better every year, will offer us incredible new insights about what contributes to human disease and how to practice medicine by retroactively looking at what's happened when people have been treated and these various data have been collected. If you want to read more about the program in the United States, we have a very informative website, all of us at NIH.gov, and we're trying to make sure we put all the information about this growing program, which is just getting off the ground. And we imagine having 1 million people recruited to the program over the next 3 or 4 years. That we think this is all very exciting. So that is what I wanted to share with you today. As you can see, it started with the Human Genome Project, but that was just the start. The last 15 years have seen remarkable advances in genomics that have taught us important lessons that are pointing us to the future that even go beyond genomics into precision medicine. I think everything we have done in genomics, starting on day one of the Human Genome Project, was about sharing the information, sharing the data worldwide, using the internet and other abilities to disseminate information, data, and knowledge. And I think you can tell from everything we've been doing in genomics since the Genome Project and going forward in precision medicine, we'll embody that same kind of collaborative, open, sharing spirit. And I hope all of you will participate in this along with the rest of us in the world. Thank you very much for your attention.
Thank you very much, Dr. Eric Green.